Not yet? Now it is. Okay.
Good.
I started recording, I think.
Okay.
So then,
Very nice. So good afternoon, everyone. Thank you for joining us. It is a pleasure to welcome you to another session of our webinar series dedicated to magnetic particle imaging. As many of you know, these sessions are normally introduced by Gabriel Pinto from Aveiro Institute of Materials in Portugal. But today it's going to be me. I am Elena Gavilán Rubio from Complutense University of Madrid, from Spain. And I am delighted to guide today's session. Just for you to know, we are recording this session and other sessions about magnetic nanoparticles and MPI, and you can find them in our website, so nexMPI.eu. So today we are going to have the webinar from Dr. Marco Giardiello, and our next webinar will be from Dr. Nuno João Silva next year on January the 12th about fundamentals of magnetism and electromagnetism. But let's continue. So it is my great pleasure to introduce you to Dr. Marco Giardiello. He's involved in nexMPI cost action project as a co-leader of working group 3 dedicated to the development of MPI tracers. He has a MPI device, so he's a precious member of this nexMPI project. Marco Giardiello is a senior lecturer in chemistry and UKRI Future Leaders Fellow at the University of Liverpool. His research focuses on developing advanced nanomaterial for biomedical imaging and drug delivery. And after studying in Leicester and Siena and completing a PhD on MRI contrast agent, he held research posts in Belgium and France, and at Liverpool his work on HIV nanotherapies led to the formation on Tandem Nano Ltd and first in human oral nanomedicine trials, and in 2022 he secured, as I said, the UK's first momentum MPI scanner. Yes, let's continue. Today, Marco, he will present basics on MPI tracer development. Marco, the floor is yours.
Okay. Hello, everybody. I'm going to share my screen. It should work now. And if I open up my
Yes, we can share see your screen.
Just need to swap it around. I didn't realize. Okay. And I will. So do you see that?
Yes.
Brilliant. Okay. Well, yeah, first of all, thanks for the presentation, the introduction. Just to quickly add that the scanner that we have here at Liverpool, the Magnetic Insight one. So yeah, it was one on a grant, like you just say, but the two, I saw the names pop up, the two colleagues that kind of run the scanner at Liverpool are also on this call. So if anybody wanted any questions about access or, how to actually kind of come over and start using it, and that fees associated and that kind of stuff, then actually a lot of people are here right now that can help out with that. But yeah, so thanks very much for the intro. So I've kind of skipped onto my next slide a little bit quick, but my talk today is going to be on tracer development, because that's the research theme that I kind of follow. And yeah, I'm going to try and give a bit of an overview of how MPI kind of works and sort of a bit of a basic context in the context of how to design tracers. But yeah, just to once again sort of flag that we do have the momentum scanner from Magnetic Insight that's present in Liverpool. This here as well, just to sort of add, so we're talking about MPI today, but that is the hyperthermia, the Magnetic Insight hyper module that's there. And we have all the software and everything to kind of do these, to do the imaging that is needed. And quite importantly, from tracer development perspective, we do have the software that helps, the relaxometry software that helps with tracer design. So I'll do the talk as though people don't know. There could be people here that don't know necessarily what MPI is. So MPI is it's a relatively new diagnostic technique. It detects nanomolar concentrations of SPIONs, so that's superparamagnetic iron oxide nanoparticle tracers. So it's kind of like a hotspot imaging. You know, what you're seeing in these images is the tracer. It's not it's an imaging that requires the presence of a tracer in order for it to work. It offers quantified real-time monitoring in vivo with no background signal. So what you see in that image at the bottom, because there's nothing magnetic in vivo, you would not see anything in the background. Where you're seeing the skeleton there, that's an overlaid CT scan. So it's very, very useful for generating high sensitivity images with no background signal. So what you tend to get when people describe MPI, how the signal is generated, it detects the nonlinear response of SPIONs under the application of a sinusoidal drive magnetic field. But to kind of simplify this, fundamentally, MPI does two things. It does them together, but it does two things. The first is it employs gradient magnetic fields to define this zero field region called the field-free point, or the field-free region, or the field-free space, some people call it. But essentially what you're doing is you're pushing two magnets together, so north against north, and the field lines would repel. So in this picture I have here, in that point where the field lines repel, you have no magnetization. So if you had a magnetic particle here in the field lines be fully saturated, here it would not be saturated at all. So in doing that, what it's doing is it's defining kind of the imaging space, the imaging voxel, so to speak. It's defining where you are going to see what you want to see. And then the second thing it does is it oscillates a magnetic field across everything, but logically, the only particles that would respond to that field are the particles present in the field-free point. So in other words, and I've drawn a very simplistic diagram here. So if you imagine sort of applying an oscillating field, do you know, I should change my pointer so you can see it as a better. There they go. If you apply a field, you know, a drive field here to this particle here, well, it's fully saturated in the strong magnetic field gradient. It's not going to give any kind of response. Whereas when that field is pushed over the field-free point, your particles will react to that presence of that drive field, and it will then give a signal. To go into that just a little bit more, essentially the MPI signal is a derivation of the Langevin magnetization curve. So what's happening here is as your oscillating field goes back and forward, and just to quickly add, there's been other webinars where this has gone into much more detail. So if you watch the other ones, they'll explain this a lot into more detail. But basically, you're flipping backwards and forwards your magnetic particle in line with that drive field. And what happens is that in that field-free point, that field-free region, the derivation of this sort of oscillating magnetic field will give you this, you know, square wave function kind of derivation of that response, which then gives you these harmonics, and the harmonics are able to be imaged. If you were to apply your field outside the field-free point, so the SPIONs in the, where I've got over here, in the saturated region, they would not be able to respond. You generate no harmonics, and you would have no image. So fundamentally, you have a space where you have zero magnetization, so you define your imaging point, and then you apply an oscillating field across that point to generate an image only in that space. So that's in simple terms how MPI actually works. Just to put in context compared to other imaging modalities, I won't dwell on this because I've realized we've already kind of, because of the delay, we've already gone over time a little bit. But it basically is, I think the number one, if you can read up on these in other kind of ways, but the number one is that it has no ionizing radiation, and you get no background signal from where you do it. I guess the downside is that it does need a magnetic particle. You do have to inject something into somebody, but generally speaking, iron oxide particles are relatively non-toxic, or much lower toxicity than other things. And there can be an effect of magnetic fields on somebody, but not, I mean, at the moment, MPI is only preclinical, so it would become an issue as the clinical scanners kind of kick in. But a lot of the other require gadolinium, for example, for MRI. Well, it doesn't require it, but it has gadolinium, which is can be quite toxic. You have ionizing radiation from your other sort of imaging modality. So MPI kind of sits with all of these in quite a promising sort of technique. So the applications of MPI, they're quite wide ranging. I mean, there's a lot of people use them for lots of different things, but essentially detection is spatially resolved to that field-free point, and magnetization is directly related to the quantity of spions that you have inside there. It is linearly dependent on how many spions are present within that field-free point. So you can calculate concentration of your particles in there, and it gives you can use this to do real-time imaging, quantified real-time imaging with no background signal. So it's very useful for things like cell tracking, perfusion imaging, drug delivery, inflammation, and vascular, as well as other things that other people might be adding. So I've just added these as an example. And just to give you, this is from a group that we actually work with over in Canada, where they did a study where they were just demonstrating the linearity of particles with the concentration of iron that you have. So you see how linear this kind of increases as you change your magnetic particle. You can see it's really bright and it goes really dark as you, well, less bright as you have your decrease in magnetic particles. So there's many applications that MPI can have. I'll go into some applications first, but again, I won't, because these are obviously not mine. These are literature ones, just to give the broad range of examples of where MPI kind of aligns. But again, I won't, because we were slightly delayed, I won't dwell too much, because you can just take the reference and you can have a look. But this is the paper that says that it was the first in vivo tracking of stem cells using MPI. And the info is there of what they did. So they had these stem cells, they labeled them with Reservist, and you can see the accumulation of the cells that they were observing. This is a little bit more recent, where they were looking at using a different type of SPION to do cell tracking of these murine mammary carcinoma cells. And you can see quite how bright those images kind of appear. So it's really kind of useful. Cell tracking is probably, in my mind, is one of the main things that MPI can actually be used for. Because of the quantitative nature that it actually has. It has its drawbacks, obviously, because resolution and stuff is still something that people are working on, but it's very powerful in that way. Here's a couple of examples for cancer imaging. This is one where it was demonstrating the EPR effect, so the enhanced permeability and retention effect. The idea that tumors grow badly. So when they grow badly, the blood vessels that kind of feed those tumors have perforations in them. So when particles pass by, they kind of filter into the tumors. And because it's a hot topic, so to speak, some people do and some people don't kind of talk about EPR as being a sufficient thing. Well, in this particular paper, they were demonstrating the EPR effects of these sort of long circulating spions. You can see them accumulating inside those tumor. And in this study, they similar kind of thing, but they do have a binding modality on that surface. Going into this from a material perspective, you can sort of see straight away that in the previous presentation, we had small particles and large particles, different surface chemistries and different types of iron mean that loads of things are going to be able to be looked at when you're doing these sorts of things. So this is just two. There's quite a lot of cancer study. This is one of my papers that I really like, because this is quite similar to what my research is. So in this one, they're using it for drug delivery, to monitor drug delivery. So they made a composite particle where iron oxides and drug were kind of buried inside this composite. And as that composite arrived in the tumor, this PLGA polymers that were there, they broke down, the drug became free, and the signal changed of the particles, because the iron oxides escaped, so to speak. And the idea was that because they escaped, they were able to kind of, and I'll go into what causes particle signal to generate, but because they escaped, it was in line with the amount of drug that got delivered. So they were able to monitor when and how much drug was delivered when this arrived, specifically in the tumor site. So this is a really interesting study from a composite perspective. And for the research that I do is very much focused on forming composite organic, inorganic materials. So I really like this paper. And then the last example I give is hyperthermia, because we have the hyper in Liverpool, and we really want to promote people coming to see what we're doing with it and what can be done with it. This is not us. This was done by this group in America. And essentially what they have here is two tumors, and they injected the iron oxide into this mouse, and it accumulated in both tumors. And you can see this in the image that you have here, in the MPI image that you have here. They then use the hyper, which can be really focused, to only warm the tumor in one location. And then they imaged afterwards using bioluminescence, and they could see that tumor depleted, but this tumor remained still present. In other words, what they were doing was demonstrating how focused that hypersystem is to only target the heating of that particle in that one location. And if you think about how small a mouse is, you know, those tumors they look far away when you think about how big the body is, but actually they're very, very close together. And they were able to just zoom in on only that one tumor. So that was really a really good study in this context. So that's more stuff that we do at Liverpool, but we haven't published yet on that. So I thought I'd show this paper here. So those are the applications, and I hope it's shown the broad range of things that MPI can be used for. So if we look at it now from the material design, you know, I've said it a couple of times, MPI requires SPIONs. It requires a magnetic particle in order for it to function. And there's major advantages in superparamagnetic particles. So atoms, first of all, have moments much, much larger than for example, a nuclear magneton that you have in hydrogen protons. Or just protons, you have to say hydrogen proton, that you have in protons, for example, in MRI. So in principle, it can be significantly more sensitive. Superparamagnetism occurs when you have SPIONs in very small size, small single domain particles with no magnetic memory. What essentially it means is, this is that Langevin function again. So what it essentially means is, as you oscillate your magnetic field, a superparamagnetic particle will be immediately saturated. It'll be immediately magnetized in the presence of a magnetic field and immediately loses its magnetization immediately the second you take that magnetic field away. It's different to a ferromagnet where you have many particles, many sort of crystal structures together. It would retain some magnetic magnetization, and therefore you need to kind of drag it back in order to demagnetize. And this is more the principle of how hyperthermia kind of works, whereas MPI relies on this fast oscillation of your particle. If you think about how quickly that can move, it will generate more harmonics and you get a stronger signal, a stronger image. So superparamagnets, what you want when you're designing them is you want your superparamagnet to be really strongly magnetized in response to a magnetic field. So what you want is here, imagine it kind of flowing through, or your field free point goes over your particle, or your SPION goes in through the field free point. You want it to just immediately like ping off its magnetization, so only the Drought field affects it. You don't want it to kind of stay a bit magnetized. You also don't really want it, you'd like it to not really be affected where it's only slightly magnetic. You'd only want it to be affected when it's fully saturated. So in other words, you want it to switch off and on as fast as you can. So for optimal signal, you want your magnetization to be strong enough to fully saturate. So it has to be quite susceptible particle. And as the field oscillates, you want this to be fast enough to fully saturate and fully unsaturate so it doesn't lag behind the drive field that you have. You wouldn't want it to kind of retain any magnetism. You want it off and on as fast as you can. You can see the signal here is related to the time, so that the time of how quickly that kind of goes. The quicker the time, the higher the signal that you would get. The consequence of lagging, the consequence of this not responding quickly, would you get reduced harmonic content. It doesn't respond, and you would get a blurring, lower amplitude signal that you're generating. So that's what you want to do when you're designing your particles: high magnetization, really fast response to the magnetic field, and that's how you're considering when you're designing what you're designing. So the processes that cause that relaxation, so how quickly it responds, there's two processes that are the main processes that cause it. And the signal is dependent on the combination of the two of them. The first one is nAL relaxation, so that is the internal magnetic moments of the particle itself. So kind of the North Pole becoming the South Pole, so to speak. So the flipping internally of your magnetic particle. And Brownian relaxation is the physical rotation of the whole particle. So it's the whole thing kind of moving. And quite as you sort of think logically, that is dependent on the size, the hydrodynamic volume of that particle, and the viscosity of the media that it's kind of in. So the thicker it is, the harder for it to rotate. You would slow down your Brownian relaxation, and the larger the particle would kind of slow that down. It's kind of logic. When it comes to nAL relaxation, it is related to the core volume, but it's also related to the anisotropy of the particle itself. So and obviously this is there's the energy barrier that you need to kind of overcome in order for that nail relaxation to actually occur. Broadly speaking, when you have, well, it's not broadly speaking, this is the theoretical that this kind of has. Much smaller particles, it makes sense that you would have a faster, slower relaxation time when you've got very tiny particles. As those parts, so your nail is faster. As those particles are, if you make bigger and bigger and bigger particles, the nail relaxation starts to become too slow. So this is obviously the length of time. So it becomes very slow. It doesn't become the dominant thing that is what the particle is actually contributing to generating the signal, and you start to be in the Brownian domain of your particle. So in principle, that would suggest to you that you would want to make your core size, your magnetic particle, very small, so it would give you a really fast signal. This is some studies that we've been doing in Liverpool, where we made a 20 nanometer particle and a 5 nanometer particle, and we looked at exactly this. We've done some theoretical stuff, and we looked at exactly that. This actually isn't my chart, by the way. That is from this presentation here, but it's quite a widely known sort of thing. And you can see that the 20 nanometer, despite the idea that would actually be slower reversal, actually is given the stronger signal. So we're kind of kicking around this point, and the 5 nanometer particle gives no signal at all. And that's because of the whole ability for it to be magnetized. So although it would have faster nail response, the moment its magnetization is too low, it's too hard to get that to be magnetized. So theory would tell you that the best core size is kicking around. If you go too high, then it won't reverse easily. If you go too small, you don't have enough magnetization to do it. And therefore, the optimum kind of size is around 25 to 30 nanometer. That's what the theory would say. It's still not really known. There's still a lot of, and obviously this is dependent on the scanner and the system that you're actually applying. In principle, if you changed your dry field frequency or change your field gradients, you could tune to a different particle. But broadly speaking, this is what the theory would actually say. But there's still a lot of work to be done in that context, trying to understand core size and what is the optimum particle, so to speak. So to consider designing the particles, you can have what you call multi-core particles or single core particles, and fundamentally they need to be stabilized. And I'm going to talk about both. I'll talk about the making of the cores, and then later on I'll talk about the making of what you need to consider when you're making your kind of protective shell for when you have your in vivo particle. The first thing I'll show, though, is this study that was done by the group in Canada again. Paula Foster is the PI over there, but this is Olivia Sell that did all this, where they took a series of commercial particles and just compared them on the scanner that they have over there, which is also the momentum scanner, and just compared them. And this is the MPI signal, the point spread function that's generated across the MPI signals. And this is kind of what you get from your, the supplier. They call it a 25 nanometer core carboxydextran shell. That is ferrocarbotran or Vivotrax or Reservis. But Vivotrax is the commercial SPION that comes with the momentum scanner. It doesn't come with the momentum scanner. The Magnetic Insight, who make the momentum scanner, also make Vivotrax. And that's pretty okay. You can see it. It's the green one that's kind of about halfway up on this chart. Cinemag D is absolutely giving the strongest signal. That is a particle that has been designed for MPI. Ferromoxetol is an MRI agent, gives a very poor signal by comparison to the others, and very low resolution. So the height of the signal is the brightness, in a sense. The width of the signal is the resolution, so how blurred it actually is. So ferromoxetol is not as good compared to the others. And then they also compared it with these micrometer-sized iron oxide particles, MPIO. So these are very large micron-sized particles. But you can kind of see the theme here has similar, they're all quoting similar cores, despite it's the structure that seems to be making what these particles are. So this is how I've always myself interpreted what the structure of these individual things are. Other people might have different ideas, but in order for the way my brain works means I always have to make things in as easy and cartoon way as possible. So I've made cartoons of what I think is going on inside these particles. Vivotrax essentially is many 4 or 5 nanometer crystals that all clump together to form around a 20-25 nanometer core structure where you have your carboxydextran on the surface. So that's what's going on in Vivatrax. Cinemag is basically the same, slightly bigger crystals, 10 nanometer, and it forms much larger core in this sort of nanoflower arrangement. And that, again, it's stabilized by this dextran, this sort of sugar sort of shell. Ferromoxetol, if you read it on paper, it's the same. 4 nanometer cores, 4 nanometer crystals, but they don't cluster in the same way. So they're not forming that sort of overall sort of core structure that is around 25 nanometer or bigger. That is why you get such a poor MPI signal. And then the massive one, these are absolutely huge, your MPIOs. So I've drawn them the same, they look the same size, but this is a micron size, so it's significantly bigger, of many particles buried inside. So of course that's going to give an extremely strong signal, but it might not give you such great resolution because it is so big. The reason why I bring this up is if you remember on the previous slide where your theory would tell you that your 4 nanometer particles are going to give you your best nail resolution. In a sense, this is like a workaround. It's an accidental workaround, but it's a workaround on that. You have many 4 nanometer crystals all clumped together to give you your overall larger particle, and then it behaves in a superparamagnetic way when you have all these crystals together. And that's basically what they all kind of are. These sort of structures. But really importantly, your shape, composition, and the manner of the clustering will affect the signal that you get. And this is demonstrated quite nicely with these commercial particles that you see. And they all have value, by the way. So even though it's a poor signal, ferromoxetol does get taken up by many cells that you have for cell tracking. It's very useful in that regard. Same with the MPIOs and all the rest of it. So although it might give a stronger signal, you can have the strongest signal ever, but if your liver just clears out your body in 2 seconds, you don't have a product, you don't have a path, you don't have something that's useful. So although it might not be very strong, it might still accumulate where you want it and all this kind of thing. So it's not all about just making the signal as strong. But this is quite interesting when you sort of look at the structures in this way. So there are various different ways of making particles, and I'll go into them, but I'll go into them in a sort of a light touch kind of way because you can read all about them. But basically, there's fundamentally quite a lot of ways, but the 4 fundamental ones that people tend to work on is co-precipitation, thermal decomposition, microemulsion, and solid gel. And the reason I've italiced them is because the only 2 that I work on in my group are the co-precipitation and the thermal decomposition method. And I would say that the vast majority of people tend to work on those 2. A lot of people do still work on the other ways because you get lots of control. But fundamentally, when you're making spions, they must be stable to aqueous media so they can be biologically compatible. And that's the part that you have to factor in when you come to decide your synthesis that you're kind of approaching. So whenever you're making your particles, ultimately you need to have them so that they are biologically stable. If you started in a co-precipitation way, so it is already starting in aqueous media, you have to consider what the stabilizers were that allowed you to be able to make your particle. And they're quite often charge stabilized, and they would not be very useful in a biological system. So you would have to do some kind of functionalization, or you do your synthesis in situ, where you have your stabilizers already present, which is how things like Vivatrax are made. So you have to consider, though, that putting them in something afterwards for your biological application. It's less of an issue when it comes to co-precipitation because and that's the reason why co-precipitation is so popularly used. All of those previous ones, apart from maybe the MPIO, were made using co-precipitation. Thermal decomposition gives you very, very controlled, very crystalline particles that are absolutely brilliant. But the problem is they're in quite often toxic organic solvents. So you will always have to do this step of transferring them to aqueous media, and that can be a problem. Because you not only do you have to transfer them, but you have to ensure that any of those toxic stabilizers they all disappear. Then you'll have to functionalize them. Then you have to try and do your advanced stuff with them as well. So if I go back to the previous, although there's many advantages of each, you must bear in mind the end game of your particles when you're kind of thinking how they're actually going to be used. So that's how they kind of differentiate between the different synthetic methods that you want to go to. But I'd say that you can sort of see why co-precipitation, although it gives you not as good signal as your MPI particles made through thermal decomposition, it ticks a lot of the other boxes in terms of actual synthetic kind of procedure. I click forward. So I wasn't sure if I was going to include this, but I've done it now, so I'm committed to it. I'm going to teach you all about nucleation and growth. So how you grow a nanoparticle. Fundamentally, when you're making, this is not just SPIONs, this is any kind of inorganic structure. You basically start usually with some kind of metallic salt. So with iron oxide, it's iron chlorides. And some kind of event happens where those salts become solid. There then is a nucleation point, and then after nucleation, there is growth. So that's how particles are made, and it's this that you need to tune when you're starting to do your synthesis. It follows the dynamics, and this is a very known sort of process, the Lemur dynamics of nucleation and growth, where essentially, as your concentration builds up, so you start off with your iron chlorides or your iron salts, but this is the same if it was gold or anything else, and they're all aqueous. And as your precipitation, or if it's thermal decomposition, your degradation of your particles kind of progresses, the concentration of your solid starts to build up, and it grows and grows and grows. The concentration of your solid sort of material grows and grows and grows and grows until it reaches a point where the concentration gets so high that there is a spontaneous nucleation, and all of those solid comes together, and you burst this nucleation of particles that begin to form. As that forms, the concentration, obviously, of your free solid ions and your iron atoms, I suppose, or your iron precipitates, goes back down. So you go below, again, the maximum concentration. So no more nucleation occurs, and what's remaining just starts to diffuse onto the surface of those particles, and you start to get growth. So you can imagine that if you had this process kind of going, and you had an extraordinarily fast, rapid, massive nucleation event, all of your material is part of these particles. So you would get loads and loads of really small, there's nothing left to nucleate onto that surface, to grow, to diffuse onto that surface. So you limit your growth. Conversely, if you had this really slow and that nucleation happened over a long period of time, you would have much more opportunity for growth to occur, and you get much more irregular, larger particles. So by tuning those dynamics, you can tune how you make your particles. So supersaturation is the concentration that you have here. You can do this by temperature. So if you have a high temperature, you might induce more growth. A low temperature would have you'd induce more nucleation. You can have capping agents to trap the growth or trap the shape of your particle. That's the other thing that can go. The solvent would have an effect, the ionic strength and the pH, because that could trigger, you change pH, it triggers the formation of your precipitation at a different rate. So you change your pH, you would have a different rate. Mixing would have an effect, because if you mix really badly, you wouldn't have a uniform diffusion, or if you mix really well, you have a wide, rapid diffusion. And then things like interfacial tension and stuff like this, the surfactants that you have in there, because you're creating a new surface. So obviously, if you're creating a particle, and you've started in liquid, you are creating a new interface, and the whole sort of colloidal system dynamics that are associated with interfacial tensions and all that kind of stuff start to kick in as well. So there's an awful lot of stuff that you need to consider when really designing your new your particles. And it's quite interesting, because a lot of people just make particles. It's like me when I cook at home. If I'm making a cake, I don't know what I'm doing. I just mix the stuff and the cake comes later on. A lot of people do that. They know the recipe, away they go, and you get your particles afterwards. And that's fine for a lot of people. They just want to have a magnetic particle to do whatever tracking or do whatever biology they want to do. But if you really want to tune stuff, you should really consider all the processes that you can affect. And I'll start with co-precipitation. I think I'm talking quick, because I want to make sure that I get everything in before the end of time. So I'll start with co-precipitation. So it involves having iron chlorides, typically at different concentrations, and essentially you induce your nucleation by adding base. And that's what I was saying before. By adding base, you can affect how quickly or how slowly your nucleation goes. You can vary your temperature, and then you will control your particle size. And this is typically how things are done. And very often it's just done in situ. You have the dextran already in your mixture, so there doesn't need to be a post-processing, because you're already in... my PhD students have just sent me a message. I'll delete that. So yeah, you already have your aqueous stabilizer on the surface of your particle, and how you control this. And I won't read them all out, but it's like I was saying before, you can change the concentrations. It will influence the nucleation density. Temperature would influence your diffusion. pH, like I was saying, influences your rate at which nucleation occurs. And then, yeah, you can change the material. You can change your type of base, your type of iron chloride or iron salts, and all these sorts of things. So there's many things that can be tuned when you're trying to create your optimum sort of particle. Thermal decomposition is a completely different thing. It's different in the sense that it's now in an organic solvent and it's triggered by temperature. So by raising the temperature of something very, very high to almost 300 degrees or above 300 degrees of your organometallic precursor, what that does is it causes the breakdown of that organic metallic precursor. It forms lumps of stuff, and then you have your diffusion and your particle grows. It's really good method for real control of what you're making. The co-precipitation is to some extent, it's a bit of a burst. It's a bit of a slow nucleation. This one is more of a burst. So when people are using thermal decompositions, they're doing all kinds of like very sort of sensitive temperature ranks and all sorts of stuff to really control what they're doing. But if you really want to read about how how it's done, this is a really good paper where they describe this process. And again, you can control your shape, your crystallinity, your particle size and everything by altering all the things that I was talking about before for the same logical reason. The difference being that you're now operating at a much higher temperature. So that is why things change. But you can really control particle shape and size and all sorts of stuff with thermal decomposition. The problem is you end with something that's in a high quite often toxic solvents that are very difficult to boil away. Because you can't, to sterilize these, you can't just evaporate them off. You're going to have to do some kind of complicated technique to make these stable and remove those toxic solvents. This is just a little bit of a summary slide for anybody who wanted to see what the difference between them are. But basically, the advantages of co-precipitation is they're very, very simple. It's why industry tends to use co-precipitation more to make different particles. But the disadvantage is low control over shape and crystallinity. Thermal decomposition is the exact opposite. You've got high control over everything, but you have lots of toxic chemicals, like I was mentioning. And this has been recorded. And if you wanted to, you could look at this link down below and read a bit more about the specifics of those synthetic methods. So you've made your core. Now you need to think about the surface. So first of all, a lot of people, you would never actually make this, by the way. This is like the everything particle where you put everything on there. But fundamentally, the most important thing right from the beginning is making sure things are suitable to be aqueous dispersions. So you need to put, this is why saccharides and polymers like PEG and PVA or different sort of shells go on those surfaces so that they can become water stabilized. That's one of the first things that you need to kind of do. The second ways that you can do that is by introducing amphiphilic polymers. So these are just water stabilized stuff, but you can have amphiphilic polymers or amphiphilic materials like surfactants where the hydrophobic would stick on that surface and the hydrophilic would extend out into your sort of aqueous sort of environment. And this is the first thing you need to consider when you're doing your sort of processing to make these biocompatible. Then after that, you can consider targeting agents like what was given in those examples before. So putting peptides, antibodies, but you can various different molecules that might go to different places. And lots of people are doing lots of things on this. These are review articles from a couple of years ago by a group down in London that really went into a big overview of the many different things that people are doing. So specifically tuned antibodies for certain areas of the body you want things to go to or to pick up to certain diseases, disease markers, peptides, and all this kind of stuff. And then finally, which is what I'm quite interested in, is loading drug molecules. So you have what's called a theranostic. So the combination of a therapeutic with a diagnostic. So you can do you remember the one before where you had the drug inside, where you can do image-guided therapy. You can monitor where drug carriers are going, and then you can see where the drug gets released. We don't work on these sorts of things, but people are looking at DNA and RNAs with this sort of things as well. So that's your kind of much more sort of complex kind of thing. And how you do this quite often is literally just mixing. You just get an antibody, mix it with your particle. Iron oxides are oxyphilic. So if you have carbonyls or hydroxyl groups, they tend to bind to the surface of particles. Or you could have your polymer have some kind of unit that you can do some kind of coupling reaction, like an amine or a carboxylic acid or something like that, to bind that to your surface. So it's opposite to what a lot of people on, like things like gold and stuff, which need phials and that kind of thing. You need it's the hard acid, hard base kind of thing. You just need your oxygen binding things. I'll very quickly just mention, because we have it in Liverpool, that obviously you're making a magnetic particle. You could further extend that and look at things like hyperthermia, which gives you an idea of making a combination particle that would be good for MPI as well as hyperthermia, and you're making these sort of things as well. A lot of people who have moved into MPI have got experience of hyperthermia agents. And then clearly multimodal imaging, because MPI will always need another imaging modality to give you that anatomical information on what you're seeing. Because if you didn't have that, you would just see the dot of the magnetic particle kind of floating in space. That gives you the opportunity to make multimodal imaging agents as well. So you don't just have the iron oxide, you can have something else there that you can also see. To do all these different studies, and then things like image reconstruction and that kind of stuff can kind of follow from that. But the most important thing is to consider your nanoparticles in vivo. So all of that stuff is great, putting peptides and antibodies and all the rest of it, but you do need to consider, first of all, the effects of electrolytes. This is one of the most important ones, because your serum is full of salt. And if you have a charge stabilized particle, which quite often they are when you first synthesize them, it will just screen out that charge. And your particles will aggregate and they won't work anymore. And then you'll just have an accumulation of lumps of your material inside the body. So the first thing you need to do is try and ensure some kind of steric stabilization ahead of charge stabilization, so that you can avoid that. So things like polyethylene glycols and that kind of stuff are quite useful for that. The second thing you need to consider is the presence of protein. So I've just used HSA to give the example of just how concentrated it is. The human serum albumin, you've got it in your blood. They can bind, if you don't have something that can kind of repel that, or something that can fight away from, or you do account for it and you're just aware that you have it there. You just need to bear that in mind while you're designing your particles, because you can have all the targeting vectors you want, but if you're just going to get your particles surrounded by loads of protein anyway, it's not going to make any difference. And so this is also the process of your body's kind of a natural immune response as well. It will immediately launch a load of proteins and antibodies to bind to your invader, marking it for phagocytosis. So it's the way it kind of deals with foreign invaders. The next one is just plain and simple degradation. When you inject something into somebody, there's loads of salts, different pH environments, different sort of redox environments. You might genuinely break down your particles, and you lose. That does actually happen a lot when you do some cells. So you load particles into cells. Inside the cells, the particles break down, and you lose your signal over time because they just degrade. And then the last one is again just your immune response. If you have a particle that macrophage can see, it will just engulf it and take it away, and then that's your particle just removed from the body. So you need to kind of it's more important, this sort of stuff, if you're making a dual kind of drug delivery thing. You'd like your particle to kind of go where you want it to go, or you want to image a specific location. But if you are actually doing cell tracking of macrophage or whichever it is, that actually can be a useful thing. So you just need to be aware of how to design your particle to kind of prevent these sorts of events, or in the case of this, you kind of want to see it. So that's what you need to sort of factor in. And then again, I got this from a review article, all of various different things that you can put on your surface to kind of encourage accumulation in a site that you actually wanted. So this is for cancer, using what I mentioned before, the EPR effect, but you can add certain things that, again, this is an example of many things on one sort of cell membrane. You will probably not target all of them. You probably just pick a certain thing to enable your particle to pass through those membranes. So that's what you're doing when you're considering your agent that you're going to start to bind to your surface. And again, I won't go into loads of detail on this because it's just me reading out what's on the screen. If you wanted, there's tons of stuff in the literature. That's actually a paper actually more about gold particles, because there's a lot of work that's been done with gold particles in this regard. But the principle kind of remains the same. So you want an agent that enables your particle to be taken up by different cells or wherever it is that you're trying to image. So just to very quickly go on about my own research, and I will go on about this very quick because I'm running out of time. Essentially, I've been working for years on making composites. So what I call the inorganic organic nanocomposite particle. And I was doing this for a long time on HIV and cancer agents and combining them as well with imaging probes. So I've worked on gold, worked on lanthanide upconversion particles and MRI agents. And I basically am now looking at this kind of thing where the cargo now is no longer a drug molecule. The cargo is a imaging agent. And we're starting to get some interesting kind of stuff. And we're looking at different structures and compositions that we are getting to monitor basically drug delivery and imaging qualities and all this kind of stuff. I'll just skip this to the end because we're gonna run. But essentially how you do it, it's very, very straightforward. You get an amphiphilic polymer. So it's in a solvent that is a good solvent. So it's a polymer that is fully solvated in a good solvent for the polymer that is miscible with water, but not, it's miscible with water, but your polymer would not want to be in water. So what happens is you rapidly, when you add this to water, you rapidly switch the concentration, the solvent environment, your particle that your polymer is in. Those polymers shrink up and they start to aggregate together and you form particles. It's not quite the same as the nucleation and growth, but you can kind of use that as an analogy. You basically make small nuclei and they all come together and they kind of grow. The reason why it's not the same is you have to factor in things like solvent expulsions and all this kind of stuff and the curling of your polymers. But essentially that's what we do. And if you add a guest to this, so if you add a drug molecule inside this, your drug would be encapsulated within the polymer particles that ultimately you get, and then your solvent evaporates away and you're just left with your polymer particle. So this isn't a novel thing in a sense, but you can really control what you're trying to do by the structure of the polymers that you have and then the condition that you do this in. And when we did this study back, this was a paper almost 10 years ago now, which is kind of terrifying. But we had SPIONs. These were commercial SPIONs because at the time we weren't making them ourselves. And we recognized by altering the conditions, we were able to put SPIONs on the surface of our particles or put SPIONs in the center of our particles. And we'd have all various different sort of properties that were kind of associated with that. And this is just one example I've been given, but forming these composites is basically what I've wanted to do for quite a long time, but not necessarily for right now. We're looking at it for MPI. We were just looking at it from a composite material perspective, and this is published in this paper here, and we got a couple of patent filings as well based on this. What I'm now doing is rather than making, well, I'm not doing it. My postdoc is doing it because she's a lot better than me at making particles. But what we're now doing is using our own, rather than commercial SPIONs. She's using thermal decomposition, and this is just the procedure that she follows to make these really, you can see she's making cubes. She's making them in different sizes. This is just a couple of the ones that she's done, but she's got tons of these. We're trying to investigate the interaction of these particles in various different polymer structures to kind of see the images that we're able to generate and the signal variation and all this kind of stuff, so that's one of the themes that we're actually working on. So we work on thermal decomposition so we can make these composites. The second thing that we've been doing is trying to manipulate the co-precipitation by making amphiphilic polymers. So rather than right now things are done with dextran, and you do co-precipitation where you have your polymer. So it'd be dextran, you change your pH and you add your iron to it and you start to generate these polymer particles. So that is the procedure that is done to make things like Vivotrax. What we're doing is altering the composition and the architectures of those polymers and various different concentrations. And what we're observing is by changing the ratio of those things and the various different structures, you do get a different signal from what is getting produced, presumably because we are making variations of, if you remember earlier on, where Cinnamag had the bigger core with many ones and furomoxetol had loads of little cores. We're probably doing that. But what's really interesting is we've done some very provisional in vivo studies with this. They give you a different biodistribution as well. So although you get a stronger signal with something, if it doesn't go in the right place, it's not really useful as a particle. So this might be absolutely brilliant, but if it only goes to the liver and doesn't go anywhere else, and it's not the liver that you want to see, then it's not necessarily as good as this one from a bio distribution perspective. So what we're trying to look at is the relationship between the two. And then the final thing that we've done quite recently, and this recently got published, was we started to just look at the calibration methods that are going on when people start to do. So I'm highlighting this because people might want to factor this in when they start making your own particles. This is the very typical serial dilution method that people tend to do when you're doing a concentration to make your linear sort of profile. So you have your material. So if you're doing a UV trace, for example, you would have your material, you take half of it out, you top it up with solvent, you take half of it out, you top it up with solvent, you take half of it out, top it up with solvent, and so on. But as we've seen, particle interaction when it's magnetic does have an effect on the signal that you get. So this is not actually necessarily going to give you absolute linearity when you have your particles, when you're using magnetic things. So we had a quick look. We wanted to see, well, I'll say quick. We had a look at what it would be if you just didn't top it up. So this here, number 4, they have the same amount of iron oxide particles, but the concentration at the bottom logically stays exactly the same. So in terms of iron mass per signal, it's not concentration that's the same. So it's not the volume that's the same. Your iron mass is the same in the top and the bottom ones each time when you're doing your calibration. But what you're doing in the bottom one is you're maintaining the interparticle proximity that you're getting. And we saw a difference. So obviously the first one here is exactly the same. We did it for Promag, we did it for Vivotrax. They are going to give you exactly the same signal. But as you start to dilute, you get a little bit of a divergence between the two, and it doesn't pass through the origin because you're not ever going to get zero amount of magnetic stuff. A lot of people who do their bio experiments will adopt this method, but actually you could be just slightly inaccurate while you're doing it. So your particles are going to interact a little bit. You may or not be bothered about this because it's not much of a difference necessarily. But when we were doing these, we were seeing up to around 10% difference when you have your particles in the you consider that versus this, they're very different in terms of dilution. And we did it with Vivotrax and Promag, and basically saw that you get a difference between materials. So it's not a one size fits all. So it's less of a problem when it comes to Vivotrax, more of a problem when you've got different particles, but it's kind of important when you look at this. But when we really saw the difference was when we loaded them into cells. Logically, if you're trying to do an in vitro calibration and apply that to what you see in cells, you are not going to be able to align them up, essentially. And that's what we demonstrated here. We can see the Vivotrax, that's your in solution calibration. If you apply that, you are way off when you're considering what you're considering. So you do a cell tracking study, you're not going to think you've got the same particles that you had. This was kind of a quick demonstration there's a lot more stuff that we could probably look at when it comes to this, but I thought I'd highlight that paper because we also recently had it published, and we got some a lot of people kind of interested in looking at that, so I thought I'd bring it up. It's a bit of a shameless promotion as well, why not? So yeah, that's the end. I think I've kept under the hour, but I've overrun slightly because we were delayed at the beginning. But yeah, I hope what I've highlighted is there's many opportunities. So this was a talk about the synthesis of particles, and I hope what I've demonstrated is there's many opportunities here for particle synthesis for materials chemists to kind of explore. You've got signal optimization, so looking at your actual core, your shapes, your multi-core sort of stuff. You've got biological optimization because you can make the best signal ever, but you need to optimize it in a biological way so that you can divert your particles to where you want to image. And then you've got your composite material for things like what I'm working on, which are things like hypothermia agents, but also drug delivery and multimodal imaging and all that kind of stuff as well. So this is all the acknowledgements. So that's everybody that's at some point been part of my group, and these are Liverpool collaborators and our collaborators over in Canada that we have, and some other people, and all the various different funding bodies, and obviously the cost network here as well. And that's that. Thank you very much. I think that's the end. That is the end. Shall I unshare? I think.
Thank you so much, Marco. Fantastic overview. Very interesting.
I don't know if I spoke a bit quick because I really wanted to make sure.
No, for me it was great.
Yeah.
Thank you so much. Do we have any question from the audience? So take this chance to ask Marco whatever question you may have. Well, Hamza has asked whether we have recorded the session.
Yeah, I
Yes,
did
we have
record it.
recorded it, and we will
Yeah.
post it on the website. Okay. If okay,
Somebody has a
so
question.
yes,
Yes, hello, I have a question. Can you hear me?
Yeah.
yes.
Okay, great. Yeah, I was wondering, so you precised all the differences according to the contrast you may have in MPI. You spoke about hyperthermia. Do you know if different type of synthesis you use result in different type of hyperthermia, magnetic hyperthermia capacities?
Yeah, in principle, the process of what generates heat is going to be slightly different to what gives you an MPI signal. So you are still oscillating, your field.
Yes.
Hyperthermia kind of relies on the lag. The way I sort of simplistically kind of say it in my head, it relies on the lag. So you want it to be, you would need to sort of pull that back. Do you remember the hysteresis loop that I was showing? You kind of want it to be separated out, because obviously pulling it back creates that energy penalty, and that's what generates the heat. So
Okay.
it is a bit of a difficult one to kind of make an optimized MPI particle and an optimized that is also optimized for hypothermia. But in principle, the synthetic methods that I was showing are the same synthetic methods for the two things. So
Okay.
as well to kind of make them so it's a one size fits all, but they are the same sort of processes. Yeah. Is that I think I've answered your question.
Yeah, no, that's a very great answer. And by the way, thank you very much for the very nice presentation.
That's okay.
I have some more question but maybe some other question, and
Yes,
or
go
I
ahead.
can, yeah, okay, go ahead. So what about the doping? You did not speak about all about the doping, and I see a lot of publication with magnesium, cobalt,
On iron
doping.
doping, yeah. So we don't. Well, we actually are looking at that, but not for. So I thought I'd restrict everything to the particles that people
Okay,
are using on the scanner. But yeah, if you
makes
dope
sense.
particles, a lot of what things people are doing is it's called blocking, magnetic blocking. So if you were to dope your particle, you make the Néel reversal really hard, and you restrict only into the Brownian. So I don't know. And I don't know it well enough to know if doping benefits MPI. I don't know if anybody else in the room might know a bit more about that. I know doping
I
more
have no
from
idea.
I know doping more from a hypothermia perspective, because it blocks out the Néel. Oh, it makes the energy barrier higher in order for Néel reversal to occur. So that's what doping does, and it's with people do it with manganese maybe, or magnesium, or the chromium as well as another one, and
Yeah, cobalt as well, I see
essentially,
a lot.
in order to do it, you blend in those salts, and they
Yeah.
as your crystal forms, they sort of dope in there. So yeah.
Yeah, thank you very much for this answer. Thank you very much.
Are there any other questions from
There's
the
Yeah,
Hamza.
yeah, yeah. Thank you for your wonderful presentation. My question is regarding the thermal decompositions, where you have two different shapes, the spherical and the cubes.
Mm-hmm.
So, do you have a better magnetic MPI signal when you compare the spherical and the cubes?
Yeah, we actually had the better one from the spheres, funnily enough. Yeah, we got the strongest signal came from the spheres. It could also, so when we made them, we're still actually studying them a little bit, so it is a work in progress. When we made them, we think that the manner in which they were made, there was a bit of an inert kind of non-magnetic shell that kind of built on top, and we need to kind of work on that a little bit in terms of actually doing them. We made this, in other words, I think we made the spheres better, is what it actually is. I think they were more magnetic. I think what happened with the cubes, so with the TEM looks fantastic because you only see the shape. But I think with the sphere, with the way we did the cubes, I think we need to work on our control just a little bit more. So the person that worked on all of that was working on making all sorts of various different shapes, and the ones that kind of gave us the best signal, we then progressed in terms of the actual imaging, and then we haven't yet gone back to optimizing the shape. So we do it in a bit of an iterative way. But yeah, so I wouldn't say that cubes definitely don't give a better signal. It's just that's what we observed at the time. But I don't know if it's because they're cubes or if it's because we had the inert shell kind of on the surface. I think it's more that we didn't make them as magnetic as they could have been, because I think not all the material was there. We're actually doing some simulation, some modeling on that as well. In Liverpool, we have I've forgotten what it's called now, the Centre for Digital Chemistry. It's got a proper name and I can't remember, but the Centre for Digital Chemistry, all these AI sort of associated with chemistry and predictive modeling and all that kind of stuff. That goes on in Liverpool quite a lot. So with somebody over there, we're actually looking at that right now to see if we can align the two things. Yeah.
Thank you.
Thank you.
Marco, is it did you check on XRD? Because sometimes with iron oxide, because I think that you produce the iron oxide and then you decompose it, is it
Yeah.
possible that you have a phase like boostite or something like this?
Yeah, well, it is possible. We didn't see it though. We just saw them still as the same phase. I genuinely do think we made spheres within an inert shell that made them look like cubes. That's what or they maybe if they're not quite spheres, they might be like octahedrals or something. Do you know what I mean? It might be a different sort of shape. But you're right. I mean, when we did them, it's been a while since they were done. So she ironically is also Spanish and also called she's called Elena, so it's different.
Oh.
She's actually on maternity leave at the moment. She'd be here and she'd answer this question way better than me, but she's not around at the moment. So we haven't actually studied them as much as I would have liked to. I think I basically put it in that slide to show the range of stuff that can be done. But yeah, definitely, if you know more about that, that would be something that would be quite good to talk about. Yeah, because we'd like to understand it a little bit more. But what was quite funny was the spheres also were a little bit bigger as well, and we wonder if it was not the spheres. I think were 18 nanometers. The cubes I think were about 14. So it might also be that because they were bigger than the cubes.
Ah, okay.
That could be why we got a stronger signal. So yeah.
Okay. Anybody else? If not, I have other questions myself, if I may ask, Marco.
Yeah, there you go. I hope I got the answers.
Yes, thank you. As I said, very interesting talk. So, considering the stability in electrolytes, so all the parameters that you said, like the formation of the protein corona, the degradation profile, the potential triggering of an immune response, and the fact that you want these particles for MPI, can you recommend like some coating, like? Do you have any coating that gives you like I don't know nice MPI signals with particles and nice I don't know stability in electrolytes and so on?
Yeah, I mean the ones that because we work on amphiphilic block copolymers, and when I worked on all my nanomedicine stuff that I was doing in the past, our HIV stuff and all the rest of it, my own sort of research experience focuses quite a lot on polyethylene glycol copolymers. So we've seen enhanced circulation and enhanced sort of things like that. And they have given so that the copolymer enables kind of the long circulation time, so to speak. So we have generated stuff that does have reasonably long circulation time, but in terms of signal, it's not been it's nothing seems to be better than dextran in terms of the co-precipitation just making the signal. So if I were to recommend, if anybody were to have a look at this, it would be, if you want to improve your sort of accumulation into, so what polyethylene glycol will do is it'll enable a steric stabilization. So the way steric stabilization works, it's not sensitive as much to charge stabilization. So you will just eliminate that problem with that. The second thing that polyethylene glycol will do is every PEG unit has 2 associated waters. Somebody told me once. And itself looks a bit, it's got an oxygen and the hydrogen is on the oxygen. Itself is water-like structure. So effectively what you're doing is putting they call it the stealth sort of PEG kind of effect of having PEG all around your surface that the immune system doesn't see. So that's why PEG is quite useful for generating long circulation times. It does, by doing that, increase your hydrodynamic diameter. So that could be as a consequence of lowering your signal because you're creating a larger overall sphere. So that could be something that's going to be an issue there. Also, the nature, the way our polymers collapse need to be tuned quite well to kind of collapse those clusters that you have so that they're tighter together. So I think it's like a massive work in progress. So all those things that you saw, the examples that I gave before, like ferrum oxetol, for example, might give a really low signal, but people have done cell study with them because Cinemag is really strong, for example. It's a really strong signal, but loads of cells find it really hard to take it up. So it's just not useful in certain ways. So there isn't really an optimum. I would say that people should think about the thing that they want to actually image and then tune it to that. I think I've given possibly the most rambling answer I could have given there, but I hope that it was okay.
No, it was okay. But
I think I shot off on various different
trying to
things as
figure
I was.
out myself this. Then this is why I asked
Do
you.
you know what it actually is I honestly think that there is never going to be a one that is the best one. So I gave those 4 commercial examples. People use all 4, so it's optimum to the thing you want to see. That's what I would say. And so yeah, I would consider it more from it depends, because if you're all about trying to improve the signal, irrespective of what the actual use is for, so you can make a really great signal, then find out its biodistribution. Then you would work on a composition that would create the larger structure, the larger kind of cluster together. But if you're really wanting to target something, you might have the end goal in mind. Do you know what I mean? So if you want long circulation, you might just go, right, we need PEG. But if you want accumulation in a certain place, like, right, we need dextran, and then you're tuning it to the end rather than at the beginning. So we've worked quite a bit on, we'll just make what we make and let's see what it does. Like almost like a bit of a sort of shotgun kind of cowboy approach to doing it. We've made this, where does it go? Right, good, and we'll make it for that then. Yeah.
Right. Right.
Yeah.
Thank you very much. Any other questions from the audience? Okay.
I'll make sure it gets recorded. So I'm going to stop recording now, so it should record. So
Okay.
at the end of the recording, this will be the bye-bye.